Hello everyone, how are you all doing? I am Suchita Nalgundwar and today my topic of discussion will be pedigree analysis. Before starting pedigree analysis, I would request all of you to subscribe to our channel to get regular updates, and your subscription is very valuable to us. So let's get started. चलिए प्रारंभ करते हैं. In the pedigree analysis, the inheritance of a particular trait is represented in a family tree over generations. A pedigree chart displays a family tree and shows the members of the family who are affected by the genetic trait. Now, before we start the pedigree analysis, we have to first know the important symbols that are involved in the pedigree. So, if you have a blank square, it represents a male. If you have a small circle, it represents a female. If you have a rhombus, then it means that the sex is not specified. Now, if the circle, the square or the rhombus is shaded, as you can observe here, it represents affected individual. That means a shaded square represents an affected male, a shaded circle represents an affected female, and a shaded rhombus means that the individuals are affected but we are not sure of the sex. Now, if a line is present between the male and the female, then it means mating has taken place between the male and the female. If there is the presence of two lines between the male and the female, then it represents mating has taken place between the relatives, and this is known as consanguineous mating. Moving ahead. So, in this case, male and the female represents the parents, whereas these are represents the offsprings. That means the parents are above and the offsprings are below. Now, if the parents are non-shaded, whereas the offspring is shaded, that means that these parents have given birth to an affected individual. In this case, they have given birth to an affected son. Moving ahead. Now, pedigree can be analyzed to determine two things. First thing, whenever you are studying a pedigree, your first intention will be to identify the dominance. That means whether the trait which you are studying is a dominant trait or a recessive trait. Now, once you have successfully identified whether the trait is dominant or the trait is recessive, then you will move on to study the linkage. By linkage, I mean that you have to understand that whether the disease or the trait that you are studying is an X-linked trait or is it an autosomal trait. Now, whenever you are analyzing a pedigree, my dear students, you must remember that the males are hemizygous. Hemizygous means the male will have X and Y sex chromosome. That means the males will have only one X chromosome, and this X chromosome will contain only one allele for the X-linked gene. For example, if I say that XC represents color blindness, if I say that XC represents color blindness, then XCY will indicate a color blind male. Try to understand, students, that color blindness, which is an X-linked and recessive disorder, color blindness which is an X-linked and recessive disorder, is not expressed just like that in female. If the female is XCX, then definitely she will be a carrier, but she herself will be unaffected. That means a single colorblind gene cannot affect the female. But as the males are hemizygous, for that reason even a single defective gene on the X chromosome can make the males defective. You must remember this. This is very, very essential in solving any X-linked pedigree analysis. Now, the points that you should keep in mind. An individual will be affected when the individual carries at least one dominant allele of the dominant trait, meaning that the dominant allele is able to express itself even in heterozygous condition. A dominant allele, as it is able to express itself in heterozygous condition, then an individual who is having at least one dominant allele will also become affected. But if the trait under study is a recessive trait, then remember that two recessive alleles are essential for the expression of a recessive trait, because the recessive trait can be expressed only in homozygous condition. So the dominant trait can be expressed even if there is the presence of one dominant allele, whereas the recessive trait can be expressed only in homozygous condition. Let us understand this individually. So let us think that capital A is the dominant allele which is causing a disease, right? So a disease is caused by the allele capital A. So if you have capital A, so definitely in homozygous dominant condition, the disease condition will show up. If it is capital A small a, even in heterozygous condition, as capital A is a dominant allele, so the individual will be affected. Whereas if it is small a, only then the alleles for the dominant trait is totally missing. So for that reason, small a will be unaffected. Now, next criteria. In the next criteria, we are saying that small a is the recessive allele which is causing the disease. So capital A will definitely be unaffected because there is no disease trait present there. Capital A small a, this is a heterozygous condition, but you know that a recessive trait cannot express itself in heterozygous condition, so capital A small a will be unaffected. Whereas small a, this individual will be affected because in this case this is a homozygous recessive condition, and in only a homozygous condition the recessive allele will be able to express itself. Moving on towards the X-linked disorder. Now if XA represents a dominant allele causing the disease, then XA will definitely be affected, because in homozygous condition both the recessive as well as the dominant character can express itself. But XAX, will it be affected? Yes, it will be affected because a dominant trait can express itself again in a heterozygous condition. And XX is unaffected because it does not carry any allele for the diseased condition. Moving on, if you have XA, that is X small a, as the recessive allele causing the X-linked disorder, then XA will be affected because it is a homozygous condition. But XAX will be unaffected because the recessive allele is not being able to express itself in heterozygous condition. And XX, it does not contain any diseased allele, and for that reason the individual will be unaffected. But whenever you are talking about an X-linked disorder, always remember, students, that a single affected allele present on the X chromosome can make the male affected. It does not matter whether it is a dominant allele or whether a recessive allele. A recessive allele present on the single X chromosome in males can also affect the male, but it is not the case in females because females have two X chromosome. So in order for the recessive condition to be expressed, both the X chromosome must contain the diseased allele. Let's move on. Generally, for the purpose of the problem, you have to remember that the individual who marries into the family in the second or third generation are not carriers, but you have to judge it depending on the pedigree that is given to you for study. But initially, in the second and third generation, if somebody is marrying into the family, we will not consider him as a diseased condition. Now, let us discuss about the key clues. An unaffected individual cannot have alleles for the dominant trait. Definitely, because a dominant trait can express itself even in heterozygous condition. So if an individual is unaffected, definitely that individual does not have any dominant allele present in them. Therefore, he is still unaffected. Next, individuals marrying into the family are assumed to have no disease allele. Already told you, anybody except for the parents or the starting founders of the pedigree, the rest of the individual who is marrying into the pedigree are generally considered not to have any diseased allele. An unaffected individual can be a carrier of a recessive allele. This is obvious because let us consider that small a, it is a recessive trait. Okay, it is an allele for the recessive trait. If the individual is capital A small a, then automatically this individual will be unaffected because the recessive condition will not be able to express itself. So an unaffected individual can be a carrier of a recessive trait. When a trait is X-linked, a single recessive allele is sufficient for the male to be affected, because I already mentioned that males are hemizygous in nature. Moving ahead. Now, this is a very important clue to solve any pedigree. A father will transmit his alleles of X-linked genes only to his daughter. A father cannot transmit his X-linked genes to the son, because the father will transmit only his Y chromosome to the son, but the X chromosome he can transmit only to his daughter. A mother, on the other hand, can transmit his X-linked genes both to the daughters as well as the son. The mother can transmit her X-linked genes to both the daughter as well as the son, but the father can transmit his X-linked gene only to his daughter. A father only transmits the Y chromosome to the son. Very, very important. So you see, let us consider that we have an affected father. Let us consider that XC is representing color blindness. So XCY is a colorblind father and XCX is a carrier mother. The mother herself is unaffected, but she is the carrier for color blindness. So the gametes formed would be, for the male, XC and Y, and for the female the gametes formed would be XC and X. Now if you go for fusion of the gametes, so you see the father will transmits the XC to the daughter and the other X chromosome comes from the mother. Then you have the father transmitting the XC to the daughter and the mother also transmitting the XC to the daughter, and in this case XC, the daughter will be affected. Now you see the father can only transmit the Y chromosome to the son. The X chromosome of the son will always come from the mother. Same here, the father is only transmitting the Y chromosome to the son. The X chromosome is coming from the mother. Moving on, so now we will be studying the key patterns observed in pedigree analysis. First, we will try to identify the pattern of a recessive trait. Looking at the pedigree that is present here, we can say that the disease must be recessive if any of the affected individual has two unaffected parents, because this is a genetic disease. Hence, at least one parent must have an allele for the disease. If neither of the parent is affected, then as already discussed, the trait definitely cannot be dominant. Now, let us talk about the pattern of autosomal recessive. So, in this pedigree that is given here, if any affected founding daughter has two unaffected parents, then the disease must be autosomal recessive, because an affected individual must inherit a recessive allele from both parents. So, both parents must have an allele. If the father had X-linked allele, he would be affected as males are hemizygous. So for that reason, students, we say that if any affected founding daughter has two unaffected parents, then the disease has to be autosomal and recessive. Now, in this pedigree, If any affected founding father has two unaffected parents, then we cannot determine if the disease is autosomal or it is X-linked. The disease can be autosomal, and in that case, both the parents can be carrier. That means both these parents can be capital A, capital A and small a. Both the parents can be carriers of the disease. The trait can also be X-linked as well, and in that case the son must have inherited his allele from his mother. The mother can be say XCX, whereas the father can be normal XY, that is not affected. So see, the parents, if the father is unaffected, that is XY, and the mother is a carrier, then there is a possibility that the son becomes diseased. That means the son will inherit the diseased chromosome from his mother. Now, identification of X-linked disease. So here we have a pedigree. So see, when an affected non-founding son, so this is a non-founding son, this is the founding son who is born to the parents. So here we have a founding daughter. So when an affected non-founding son has two unaffected parents, the disease must be X-linked recessive. The father who is marrying into the family, we will consider that he is not having any diseased allele. So the son inherits an allele only from the unaffected mother. That means the unaffected mother should have been XCX. That means the mother should have been the carrier of the disease. So you see, the father is XCY and the founding mother is XX. So the founding daughter is XCX and she marries a normal man, but she gives birth to an affected child, meaning that the affected allele has been transferred from the mother to the son. So that is why the son has become XC and Y. I hope it is very clear to you. So a male cannot be affected by a single autosomal recessive allele, but it can be affected by a single X-linked recessive trait. Now the patterns indicating dominant trait. A disease must be dominant if every affected child of non-founding parents has an affected parent. So you see this pedigree. Dominant trait can be easily recognized by no skipping of generation. You will not be finding any skipping of generation in a dominant disease, right? So you see, first let us talk about autosomal dominant. So when an affected son of non-founding parents has an affected father, the disease must be autosomal dominant. So you see that here the non-founding son has an affected father, then the disease has to be autosomal dominant because the father will not transmit the X-linked allele to the son. So the disease can't be X-linked dominant. Now, moving ahead, when an affected daughter of non-founding parents has an affected father, we cannot determine whether the dominant disease is autosomal or it is X-linked. This is because the affected father can either transmit an autosomal dominant allele or an X-linked dominant allele to his son. So you see, when the non-founding daughter, this is the non-founding daughter, is affected, whose father is also affected, you cannot say whether the disease is X-linked or whether it is autosomal, because the father can transfer both the X-linked defective gene or the autosomal defective gene to the daughter. Now, we have the question that was given in AIPMT 2015. So you have to study this pedigree and you have to tell whether this pedigree is X-linked dominant, whether it is autosomal dominant, whether it is X-linked recessive, or whether it is autosomal recessive. The first thing that we observe in this pedigree is that the father is affected and both his daughter as well as his son is also affected. So now you have understood one thing clearly, that the father cannot transmit his X chromosome to his son. That is simply not possible. A father cannot transmit his X chromosome to his son. So the first clue that you get here is that this disease is definitely autosomal. This disease is definitely autosomal. Now you look carefully in generation 2. In generation 2, you see that some of the offsprings are affected, whereas some of the offsprings are unaffected. In the generation 3, again you find that very few of the offsprings are affected, but majority of the offsprings are unaffected. Again there is generation 4 where the offsprings are affected. So this is a pattern which is generally observed in a recessive pedigree. This is a pattern that is generally observed in a recessive pedigree. So this pedigree that is given, it is the example of an autosomal and recessive pedigree. So students, that is all for today's lecture, and my homework for all of you is to find out the genotype of this pedigree. Can you please mail me in suchitanalgundwar@gmail.com all the genotype of this pedigree? I have already given you the answer, but let us keep it as a suspense. What will be the genotype of this entire pedigree? I'll be waiting for your answers. So that is all for today. Have a great day ahead. Thank you so much.